My name is Jawad Hashim, and I'll be conducting this workshop on BioRender, which is a web application for creating biomedical illustrations. Let's get started. This is an example of illustration created in BioRender. So BioRender can be used to create fairly complex biomedical illustrations, are fairly rich in detail, but also are aesthetic, pleasing to look at, with a good layout, nice graphics, color themes, and combinations. So it gives you an edge when you're creating illustrations, and it's fairly easy to use. So here's an example of an illustration or an image that was published in a fairly high-ranking journal. So it's it's a research article illustration that was not created in BioRender. And I'm just going to make it a little bit interactive. So are there things that don't look quite right to you in this illustration? Can you point out some maybe design issues? And those online can maybe type it also. Maybe if you can speak up.
The quality of the images.
Yeah, the quality. Yeah, the labels.
Some background.
Yeah, there is some background dots, so you can see that these smudges. So they probably printed it and then scanned it. You can also see some gaps here between the lines. The colors are not matching. The curve of the arrow, you can see this, it's not aesthetic. Okay. And just compare this with something created in BioRender. So, you can tell the difference. It's the lines, the fonts, the colors, they look like there's a unifying theme. Here's another one. This is again published in a good journal with, the graphics are not too bad, but what would you criticize in this image? What doesn't look right to you? And people who are online can also type. For one thing, the enzyme looks like a Pac-Man. It's a bit cartoonish. Very basic. Yeah, I'd agree. It's not too bad. It was published, but it's something that doesn't look very professional. It's hard to put your finger on it. But again, compare this with something from BioRender and you can see the difference immediately. Here's another image. This is not too bad, but what's the problem with it? It's hard to follow, but also it's fading. It's like hazy. It's not clear. Yeah, it's and compare this with something from BioRender. So again, the advantage with BioRender is it gives you a vector output. So all your images are like razor sharp, extremely clear lines. There's no fading or fuzziness that you see with scanned images or captured images that you do with a screen capture. So again, very sharp images, and then you can output at extremely high resolutions without loss of detail. Here's another one that has not been created in BioRender. What do you think of this one? Any issues? It's not too bad, actually. It's fairly professional. Any problems? Or the colors are off? Yeah. Anyone else? What do you think of the mitochondria? Yeah, I mean, look at the septae in the mitochondria. It doesn't look right. Okay, even here, the lines are. It looks like somebody has drawn this with a mouse. So it does not look professional. And believe me, journal editors are. Yeah, it's not well drawn. It's a bit chaotic. Also, you could say that it's a lot of numbers that you will have to refer to from the legends to understand this figure. And compare this with something from BioRender. Again, you can see the amazing clarity, the icons, the kind of the flow. It's really different. One more example again, and this is now better still. Again, it has those Pac-Man icons. Doesn't quite look professional. I'm sure people put a lot of work into it, but in a fraction of time using the icons from BioRender, you can create a much better, much more professional, and I would say more impressive figure. Again, look at the colors, difference in colors also. So yeah, BioRender, again, it builds on that, this gap in creating biomedical illustrations. People used to do it manually using shapes from PowerPoint, and it's very hard to get the right proportions, the right color combinations, whereas it's much easier in BioRender. Something, and if you like it, you may start using it. So one thing I want to mention is that design skill is part of your learning. So as graduate students, your design skills should be at par. You should not submit work that is sloppy, it doesn't look good, it's not aesthetic. So it is not just that you need to be good in your science, which you must be, of course, but you need to be good in your design. And design qualities include layout, color choice, combinations. So what it really boils down to is craft. The craftsmanship of any product that you produce, you put your name on it. Try to make it as best as you can, and don't spare any amount of time to improve and to perfect it. Okay, so I've divided my workshop into 3 parts. What is BioRender? Then we'll look at some templates, and then we'll end with some hands-on practice. So let's get started. BioRender is basically a web application. So you can access it in a browser like Google Chrome or Microsoft Edge or Firefox. And you don't have to download or install it. You just sign up on it. And it works nicely. It's the big advantage, it really, I think, has taken biomedical illustrations by storm. There's a lot of people who really love BioRender. It has made a big difference in their lives, you could say. You can see some of the comments on Twitter. I think the biggest advantage with BioRender is its icon and template library. So you don't start with a blank canvas. You can start with a template, and you have some amazing icons that have been professionally drawn. And you can look at the categories and the range of templates, and it includes pretty much everything in life sciences. But I think they will probably expand it to include more and more disciplines. Okay, so there are some generic templates also, like this Gantt chart, so you can create charts, graphs as well in BioRender, apart from biomedical illustrations. Okay, coming to the pricing, there is a free version that you can just sign up and use for educational use, but it is fairly limited. You can only create 5 figures, and you cannot publish to a journal. So that really limits its use. It's really, I think, to try it out rather than for serious use, because you really want to use to create illustrations for your theses or journal articles that you want to publish. So maybe the free plan is simply to get you started, and then if you like it, you can start with the individual plan, which is $35 a month. One thing you could do if you just want to use it one time, to make a one-time payment for 1 month, and then you can cancel your plan. But you cannot use the free plan to create an image for publication. So the other thing that I found a little bit heavy was that all figures must be created with BioRender. That, it's like, you want to write with every presentation created with PowerPoint. So it's an advertising for them, but they want it. But one thing they say is you can write this anywhere you want. It doesn't have to be next to the figure. It can be in the acknowledgements or even in the methods section. So if you put it right next to the figure, it's a bit distracting. If you use a template, you even have to cite it. So in your references list, you will have to cite BioRender. And again, I thought this was a little bit of an overkill, but this is a requirement. And this is even after you have paid. So, you still have to cite it. If you don't use a template, then you can just put it in the acknowledgements. That's enough. So here is a comparison of the licensing usage, but even in the free, they are pretty generous. They allow you to use it for conference presentations, teaching slides, even your personal website. So you can certainly use it for your presentations as a free plan. The problem is still that they still have what is called a watermark at the bottom right of each image, and you're not allowed to remove it. Again, it looks very distracting. So I think for any kind of serious use, you really have to buy or make some payment. And finally, I think which is the biggest issue is if you want to export your illustration, the free version only has a 72 dpi, which is just enough for web, maybe also for PowerPoint presentations, but really not enough for a journal article figure, because then you have to export at 300 dpi or 600 dpi. And if you don't have that, then journals will not accept it, and so you really have to pay if you want to use it for research articles. Okay, so we covered the basics of BioRender. Let's move on to what are templates and icons in BioRender. Templates are, I think, the key forte, the main selling point of BioRender, and they put a lot of work on it. It's not just icons, but it is a combination. And sure, you can do all that in PowerPoint using rectangles and circles, but it will take you a long time. The proportions will not come out right. The colors will probably not be as good. And there's a whole host of features that I'll show you that really makes it worthwhile, looking carefully at BioRender. Templates, come in with kind of a pre-made flow of different maybe experiments or studies that you can use and start modifying. So here's is a classic example of, say, a laboratory experiment. This is a template, and you can just change the sample text, put in your different stages. You can immediately see it looks very professional. The layout, the proportions, the text, the colors is amazing. And you can just get a really good start. You could make this using icons yourself, but that would probably not come out right in terms of proportions and spacing. Would you agree with that? Have you tried making illustrations in other programs? PowerPoint. Anybody else uses any other program to make illustrations? Yeah, Illustrator is very difficult to use, probably used by professionals. Is there something similar for chemistry? Chemistry, I'll show you some icons. So this, you can really use it for chemistry as well. So you're in the right place. Anybody else online who's used another program to create images or illustrations? Okay, so you have other templates. I'm going to show you lots of templates, and my goal is to familiarize you with the range of templates so you can see what's useful for you. There are literally hundreds of templates in BioRender, and you really can get a head start by using a template. So pathways for people from biochemistry, you can show cellular, extracellular pathways. You can compare pathways in experimental and control conditions. So these are all ready-made templates. You can just start using them and start modifying. And you can also use it for clinical sciences. So not just for lab experiments or biochemistry, but also for clinical sciences. And a lot of journals, like especially if you're writing a review article, it's very nice to have these templates that you can modify, and then you kind of produce a nice figure that you could also use for your graphical abstract or for your presentations. More templates, and some of these are fairly sophisticated templates that you can see the amount of work that has gone into them in terms of layout, and some of them are commonly used experimental designs. There are some simple templates also that you can use. They are fairly generic, but again, the layout, the fonts, and the overall design, it's very professional, and you would be better off using these than creating them on your own. Of course, it's not just human or animal models. It's also the full range of life sciences, including agriculture and botany. Okay, moving on to icons. Again, I want to familiarize you with the range of icons because there are literally hundreds of icons in BioRender, and you have a lot of choices. Icons come in two varieties. Some of the icons have this purple square with a circle and a triangle that shows that the icon is a grouped icon. So it can be broken down and individual parts can be changed. They can be rotated, the color can be changed, the outline can be changed. So you have two antibody icons. This one is one shape. So you cannot change the individual parts. You can just change the color overall appearance. But this icon comes with an edit group button at the bottom, and you can change the different parts of the antibody, change its color. There are, of course, a lot of cell types, including epithelial cells, immune cells, and blood cells. And this is not the full range. This panel is showing just the top parts. You can scroll down, and some of them have dozens of icons in there. Antibodies, of course, a wide variety of icons for that. Receptors and ligands. And again, you can see the amount of work they have put into in creating these icons that you can use in your illustrations. Transporters, so different proteins showing cellular signaling and your pathways. Chemistry, somebody mentioned chemistry. So you have a wide range of molecules from amino acids, nanoparticles. You can use these generically. Maybe showing antigens or other kind of particles, structural formulas that you can modify and put labels on, and of course mice, all different and in different icons, bacteria for people in microbiology, and mammals. So if you're talking about zoonotic infections. And each icon, like a mouse icon, can be changed in terms of color, and of course size, rotated, transparency. Saturation is the brightness, level of brightness of colors, and glow creates a nice glow around the icon. So it would, if you want to show something is activated, you could add some glow to it. For text and shapes, there are preset styles. So, nice professional color schemes, shapes that you can, the fill shape and the stroke, which is the line around the shape of the shape, you can change those two as well. So this is from this edit menu button, will open up the styles panel. Brushes is something in the premium version. It's not in the free version, but it is again something which is like a little bit magical. You can create like cell walls, cell membranes, vesicles, and you can expand them. So as you lengthen them, it adds new molecules to the length. So it's not stretching them. It's actually, and I'll show that to you. And you can change the shape precisely as you want it to maybe showing some kind of exocytosis. Lines come in a nice, variety with arrows, lines, inhibitors, neurons, circulars, lines. And there's some very nice features, including shading and, the tapering lines. It looks very professional. And then you have, circular lines that you can show maybe some kind of pathway or feedback. There are custom shapes, and again, I'll show them to you also. These have points on them that are called handles, and if you move them, it kind of like a squishy shape. It reforms nicely, a little bit like Play-Doh or dough. It has a nice, you can shape it to overlay it over some other icon. A lot of arrows also that you can modify color, have color gradients, rotate, increase in size. Text shapes, this is separate from insert text. If you use insert text, it's just text without any border. But text shapes from the insert shape menu, you have these borders, a little bit like text boxes in PowerPoint. But again, the layout and the positioning of the text, it looks very professional. You'd really have to work hard in PowerPoint to recreate that. And then all of them, come with options to modify the shape, the text, and then a lot of preset, professional presets. And then there is a floating vertical toolbar on the right side of the window of BioRender. And it shows there is a zoom to fit. So you can just quickly click this top button to expand your work area to fit your screen. There's a zoom in button and a zoom out button and a zoom slider. And there is, you can click on this 92% number. It gives you an options for zoom presets. So if you it's 50% zoom. You can hide the ruler, which is around the border of the screen. There's a ruler. And you can show grid. The grid can be modified and you can snap objects to a grid. So create a consistent layout. There's a preview button which will just show the final image without any toolbars or the surrounding interface of BioRender. And then there's a view as grayscale button that you can use to see if some of the elements are becoming invisible if they're printed in black and white. So you have to, like most of the journals now allow color printing because they're online. But if they have a print version or if they print a diagram as black and white, or sometimes people will print your PDF using a black and white printer. And you want to see if it will, come out okay on a black and white printer. There are lots of tutorials for BioRender, some short ones and some like an hour-long tutorial. There are full webinars, like 48 minutes, 51 minutes. And you can see here, Shiz Aoki, who's the co-founder and CEO of BioRender. She's a professional illustrator, and I'm sure she's kind of the brains behind all this, especially the amazing icons and templates. Another feature that I will not be discussing today is posters. So when you click on the Create New button, you get the option of creating an illustration or a poster. So if you're going to a conference and you want to design a poster, you can certainly use BioRender instead of PowerPoint. Comes with nice layouts, and again, you can take advantage of all the icons to create a very professional poster. I should also mention that you can use BioRender for grant applications. So if you create a very nice diagram using BioRender, you're probably increasing the chances of success of getting the grant. And I think they will add more options, including presentations. Another feature that I will not discuss too much today is, proteins. So you can import protein structures here. I've put in the code for hemoglobin, and you can rotate, zoom in, zoom out, and show different renditions of a protein, such as lines or surface. So again, very nice if you're doing work with protein molecules. Okay, so before I do the live demonstration, I just want to show you like a typical illustration or an image from a high-ranking journal. So it's not just BioRender image. There are bar charts and chromatography and all sorts of imaging and experimental models. So this typical illustration has like 1, 2, 3, 4, 5 panels, and even in one panel you have like 2 graphs. Like panel B, they have like 1, 2, 3, 4, 5 images in panel B, and it's just one figure. So if you're aiming high for a top-ranking journal, this is what you should aim to produce. And you will need graphs from, say, Excel or SPSS, plus your other images, plus your BioRender to make just one image. So it's a lot of work, but BioRender can help you with that. And then I'm doing a workshop on creating graphs later on that we'll show you some techniques for creating high-quality graphs. Okay, so that is an overview of BioRender. Before I do like a live demonstration, hands-on practice, I'll pause for any questions or comments. Okay, so what I'll try to do now is to recreate this figure. This is again not from BioRender. This is showing food allergy. So the inflammatory cascade when somebody eats food that they are allergic to. So there's food allergen being presented to the gut epithelium, and what happens subsequently with the inflammatory pathway. So not a bad diagram, I would say. They've worked hard on creating basophils and mast cells by hand. Okay, so this is a typical BioRender screen, and I've logged in. You can see, gallery, kind of the home page. And if you just start with biorender.com, this is the screen you will see. And if you click sign in, then that will take you to that page. There is, of course, all other pricing and other features pages. But, if you are in app.biorender.com, this is what you will see. And we can start. Really, the starting point is this. You can create new, click on illustration or poster builder. For now, we will stick to illustration. And this is the starting screen with a blank canvas. Here on the right side is the zoom controls. You can click and show ruler or hide ruler. You can show the grid or hide the grid. And then there's, the preview button, etc. And if I click on this reset zoom, it will zoom to what, is optimal for this screen. So this, on the left side are your icons panel and then the templates. So these are all the templates and the brushes that I mentioned. You know, these are, you can use it for epithelial lining, vesicles. You can have favorites, and then you can upload your own images to include in the illustration. And then the protein builder, you know, I'm not going to talk about that. You can, you know, import a protein structure from this. Okay, so let's start and create that figure. What we want to start with is, and it's a good point to start with, is a brush. I'm not going to use a template in this case because I want to show all the features of BioRender, but I would not recommend using a blank canvas if you can find a nice template. But for now, because this is like more like a workshop, I'm going to use a brush. Okay, and what we want is an epithelial lining of the gut. So, you know, you can just scroll down. There's lots of brushes for nucleic acid brushes, membrane vesicle brushes, cell brushes, so we're getting closer. Okay, we could use this. If I just hover, you can see it's a columnar cell brush with nucleus in the center. Not bad. Cuboidal. We're really looking for gut epithelium. So here we are. So enterocyte brush. So let's see if we search with the keyword gut epithelium, will we get enterocyte brush? We're getting some other epithelia, but not that enterocyte brush. So what I think the keyword search is not bad. We have something here. Oh, okay, we have our enterocyte row. So the lesson I want to share is that, you know, you want to familiarize yourself with the range of icons, just play around with it. You also want to scroll down the list. But you can also use this search feature, which is pretty good. You know, it uses synonyms and tries to guess what you're looking for. So we'll just go ahead and click it once. One click is sufficient. You have to wait for maybe like 2-3 seconds before it comes in. Don't double click, and don't become impatient by clicking it several times. So let's put it here. And now I'll show you some of the magical part of using brushes. I'm going to expand it laterally. Okay. And you know, this one, it is stretching the icons. So this is an icon, not a brush. And it doesn't look quite right. Now let's try it with a brush. And we'll use the enterocyte brush. So I'll just scroll down. And here it's cuboidal cell brush. Here's the enterocyte brush. So this is not an icon. This is something special to BioRender. And this is a brush. So now I'm going to stretch it, and you can see something magical happening. You know, you're getting more enterocytes being added. So it's not stretching. It's actually, you know, it's like there's cell division going on. Can you see that?
Yeah.
Okay. So, you know, I'm going to use this one. I can just click on it and hit the delete button. So some keyboard shortcuts do work in BioRender, including Control-S for save and Control-C for copy, and then Control-V for paste. So if I want to duplicate this icon or brush, I can just select it once, click on it once, press Control-C, and then press Control-V. So it has pasted another one, kind of slightly, you know, off center from it. And now I can, you know, place it. You can see this red alignment line. It's a nice alignment. This is features now available in PowerPoint as well. So alignment line comes in magically. And let's try and do a third one. And you can see here the alignment line comes in, and you can see these purple alignment lines on the right side that's showing that all three figures are equidistant. So that's very useful feature in creating a nice layout in BioRender. Otherwise, it looks like they're spaced apart a little bit unprofessionally, with varying spaces between figures. So I'm just going to delete the extra ones. So we have nice gut epithelium. I want to put in some T cells. So I'm going to search in the icon for T cell. And here we are. I'll just hover over it and you got a T cell. If you hover on this one, it has a nice glow on it. It's an activated T cell. This is a T cell with a T cell receptor. And you can see here this purple indicator below on the lower left part of the icon that shows it's a grouped icon. So if I click on it, it comes with this edit group button, and then I can start modifying different parts of it. The receptor part, I can change the color of the receptor, and the cell, I can change the cell color as well. So that feature is not available in the standard T cell icon. Okay. So I'll click outside. So this standard T cell icon, it is not a grouped icon. It is just, it allows a limited set of modifications, including color presets and some transparency, saturation, and glow. Okay, so I'm going to delete this. So we have our T cell, and remember it became activated. So I'm going to get an activated T cell. And I'm going to use a curved arrow to connect them to. So I'm going to click on the insert line, arrows, and look for a nice curved arrow. And then click and connect this. And you can see the handles inside the arrow. I want the curve to be this way. So this screen can quickly create like a cellular, activation. I want to add some text, so I just put a heading text over here. Just click once. You don't have to drag to create a text box. So maybe like inflammation. And remember, we have, the gut intestinal lumen. So I'm just going to use a slightly smaller subheading and put it over here. Just one click and put okay, and I can resize this by clicking outside and then clicking on this. Now it has kind of grouped this. So if you want automatically, I'm going to click edit group, and then I can go back and edit this. I'm going to make this, I can increase the size, or I can make it into one line by dragging on this handle. Okay. Let me change this to like a gray color. So I can click on this edit button here, change the text color to maybe like, let's use this bluish theme. And I want to use the same style. So I'm just going to click on Control-C and then Control-V to create a copy of this label. And then I want to align them together, but I don't want center line. I want a left align. So let's see, I'm going to double click on this text to change the text. Okay. So I'm going to click one and then click the other by holding the shift key down. So now, I can align them. I can click on this align icon here and align left. Okay. So it's already actually aligned left. So now, you have a nice way of showing, the structures where you are. If I want to show some food allergens coming and hitting the gut epithelium, let's use the nanoparticles icon library. You can type, let's try antigens first. Yes. What undo? Yes, there is an undo button. You can see it right here. So you can easily undo and redo. So I like this. This is not a bad antigen spray of spheres. Just one click. And it appears after a few seconds. So I'm going to make it smaller. And I just rotate it a little bit. And then I want a small label saying food particles. So for the label, just one click. Food allergen. And I'm going to bring it closer to the food. Okay. It looks a little bit small for to me, so I'm going to increase the font size a little bit. Okay. We wanted a dendritic cell, right? So here we have some dendritic cells. This one has nice squeezing tentacle there. So I'm going to use this, rotate it a little bit so it squeezes between the epithelial cells, trying to reach out. And I want a label just below it. I'm not going to use the labels with the borders around them right now. I'm just going to use a small label. I'll use the paragraph text. Again, one click below where you want it. And, move it around. It's up to you where you want to place it. Okay. Again, for some of the the cytokines, I'm just going to use, maybe because those are important, we'll use the paragraph text here, again, maybe IL-2. Okay. And remember that figure had a photo of a colon or the gut. So I'm just going to type GI tract. And you have lots of options. I'm just going to take this one and make it a little bit bigger. I don't want this mouth part and the esophagus, so I'm going to crop it. For that, I can use the crop, add crop. And here you notice you can have a square or a rectangle crop or a circle crop or a very customized crop. So I'm going to let's go with a rectangle crop. Okay. And it's not bad. And then I wanted a zoom figure, like that rhomboid figure, a rectangle that's zooming in from the colon. So I'm going to click on insert shape, some shapes, and this one is like a rhomboid, will rotate it. I can use that one. I'm just going to rotate it in this direction. Make it bigger. Okay, and I'm going to push it behind the colon, the gut. For that, I'm going to use the arrange order button and send backward. Okay, and notice that again, you have some nice keyboard shortcuts for that as well. I want to color it, so the fill. If I click on the fill button, I have lots of options. I'm going to choose something that matches the theme. Or I can use a preset here, so going with the yellow theme. Also, I want the zoom to show some kind of gradient. So I'm going to click on the fill and then click on gradient and add a dark color. I click on this gradient stop and add a dark color. So again, it's maybe no, I think I want this to be white and this to be the dark one. And I don't want a border, so I'm going to click on the stroke and make it transparent. Okay, the color is still not, and again, you can see here, there's another handle with like a brown circle that you can use to adjust the shape inside. It's not too bad, but still a bit too dark for my taste. I'm just going to make it a bit lighter. I can, reduce the, move the slider here and kind of make it lighter. I'm losing a little bit of the gradient then. So this one, or I can use a preset gradient also. So you can also have a radial gradient in which you have a circular pattern. Okay. You actually stick with a preset here. Okay, so that's enough for this figure. Of course, you can do a lot more. I think you'll agree that it looks better than the one that we saw in the journal. Okay, I'm going to do one more figure as in practice. Maybe this time I'm going to just go ahead. Sorry, how to save it? You can just click on this untitled and change the title. So it's, we have food allergy, inflammation. Wait. No, and then you can click on save, Control S for save, or just click the save button here. It just tells you it's saved a few seconds ago. And you have keyboard shortcuts that you can use in BioRender to speed up your work. Any questions at this point?
Is it possible to change the size
Of
the canvas?
Size of canvas, yes, you can change the canvas size from here. So you click on canvas size and you have some presets of ratios. You can choose your unit from inches or centimeters. You'll have to look at the journal requirements. Typically for a single column, your width should be around 8 to 9 centimeters. For a double column full page width figure, the width can be like 18 centimeters. But really you have to check with the journal, or if you're doing a thesis and you're not constrained, then you can just stick with the default canvas size. Remember, when you export, choose the pink format because it kind of does a better job at keeping the sharpness than the JPEG. Okay. And go for the highest resolution, like 300 dpi or even 600 if you want to. So you want to adjust the canvas size before you export it. after exporting, you can of course resize the image in your FontView or other imaging program, but then that will kind of maybe add some fuzziness, because whenever you change the size of a canvas in an image program, it interpolates the pixels. So the text will lose the sharpness. So if you want to change the size, better to change the size in BioRender before you export it.
Is it possible to change the size after we created our?
You can, but then you will have to rearrange, so let's say if I increase this to 20, sorry, I can only go to 16. Then, you have to rearrange the figures because you have got some extra space now.
So
it will not be rearranged automatically. Yeah, you have to, because you have it's not just in one dimension, it's the arrangement or the relationship between individual icons. You have worked hard, so it cannot really rearrange it. You have to do it yourself, but that's not too hard. So you can move this over here. So you have more space now to do things. Okay. Any other questions? Any questions from our online? Okay, let me quickly show one more figure in a few minutes, and then we can close the workshop. So I'm going to go and file, new, and then illustration. Okay, I haven't saved it, so I want to save it first. For, let's say you've done an experiment on the rat brain or mouse brain, and you want to show some areas on it. So the different kind of work than, say, biochemistry or a biochemical pathway. So let's search for mouse brain. So, you've got a lot of different, views and sections. So you have a lot of choices. I'm going to choose this slightly rotated one. Okay. And I'm going to make it bigger. And if I click on it once, then it brings up the edit options. I'm going to choose a lighter colored version. And maybe you can even make it a little bit more transparent, even lighter. If you want, you can add a glow to it. It's more for review articles, you have these nice glow. If it's more for experimental figure, then you don't want that glow. And let's say we've kind of made an injection to this part of the brain. We'll insert a circle. So just one click, wait for a few seconds, and then you can drag. And as you can see, you can draw an oval. But if you hold down the Shift key, it becomes a perfect circle. So I'm going to hold down the Shift key. And let's say this was our injection site. I'm going to make it a little bit red to show this was an injection site. Stroke, I'll make it or make it like a light red to kind of give it a glow. Okay. And then let's say we found there's some areas that were affected. So I'm going to insert a shape, and I'm going to use a custom shape, which is like malleable. It's it can be reformed. So let's say the frontal cortex was affected. I'm going to draw the shape. I can rotate it. I can resize it. I can use these handles to kind of match the contour of the brain edges. So, it looks, it matches the cortical areas. And then I'm going to show that this is an injured area, maybe with this color. I want an arrow, again, a curved arrow connecting this. Let's say if it's inhibiting, I'm going to use this inhibiting arrow. So just okay, and then give it. This is an elbow connector, so I'm going to use an S-shaped or a curved one. So S-shaped gives me most flexibility. So now, you know, it's you get a curve. Okay, so it's showing some inhibition going on. And I can duplicate this shape, Control C and then Control V, and maybe show some effect on the temporal lobe. And then again, just modify it to match the contour of the brain. And again, insert an arrow. Showing that our injection site has affected this part as well. So, okay, and then, you know, you can have show an injection icon. Waiting for the icons to load. Yeah, I mean, it's you know, it's a schematic, so it's, you know, it's showing that, you know, this is the point where we injected the drug or molecule. So again, so I'm showing some icons and features of BioRender that can be used in experimentation. Okay, I think I'm going to stop at this point. Any other questions? Okay. We can't go ahead and try and create an image in. If you can log in for free, you just sign up for free using your university email and try to create some images, and I can help you if you're stuck or if you want to know some features. So this, you know, we come to the end of the workshop itself, but I'll stick around if you have any questions or if you want to show something that you need help in. Thank you very much for attending, and I hope you enjoyed the workshop, and look forward to seeing you again.